Welcome to this new session of Transplant International Podcast.
Hello everyone and welcome to the Transplant International Podcast, where we explore innovations in organ transplantation and the science driving our field forward. I'm your host, Nina Pilat. I'm the Deputy Editor-in-Chief of Transplant International, and thank you for joining us for this February episode. Today, we are looking at two major topics from our new issue. First, our annual Editor's Choice. It's the top 12 most impactful papers in clinical transplantation in 2025. And second, a narrative review on the fast-growing role of machine learning in lung transplantation. These papers together give a panoramic view of where transplantation is heading, scientifically, clinically, and also technologically. To discuss these editorial highlights, I'm joined by Dr. Antonio Citro. He's Editorial Fellow of Transplant International and a researcher at the Diabetes Research Institute in Milan. Antonio, welcome back to the podcast.
Thank you, Nina. It's really a pleasure for me to be here. And I think these two papers that we selected actually complement each other very well. Indeed, the editorial choice maps where the field has moved clinically, and the machine learning review looks at one of the methodological forces that is increasingly shaping, in my opinion, how we generate and interpret the clinical knowledge.
Yeah, thanks. So before we begin, we would like to state that the views expressed here, they are our own, and they do not necessarily reflect those of Transplant International or research. So let's start with the editor's choice. Our editorial board selected the 12 papers we believe are most likely to shape the clinical transplantation moving forward. This year's list spans xenotransplantation, immune tolerance, stem cell-derived tissues, machine perfusion, oncology, infectious diseases, immunology, pathology, and lung transplantation, so all the major fields. And there are two xenotransplantation papers heading the list, the landmark pig to human heart and kidney xenotransplants. Antonio, why do these lead the list? I mean, xenotransplantation, it's a niche topic. If we look at clinical transplant medicine, still two papers made the list of most impactful papers for last year. So what's the impact for the field?
Thank you, Nina. So I'm really glad to speak about xenotransplantation because it's a field that I like a lot and I'm working also with. Because these two papers, because they represent, I think, a qualitative leap forward, not just an incremental step. These are the first comprehensive clinical reports of transplantation using multi-gene edited porcine donors, organs into living human recipients. What the gene modification strategy achieved is something really important. They're knocking out 3 carbohydrate antigen genes responsible for hyperacute rejection. Was to essentially disarm the most immediate and lethal immune barrier. Neither patient survived long term, but this is not how we should measure the significance of these cases. They provide data that we simply did not have done before. The multi-omics analysis, indeed, of the kidney case alone will inform the design of the next generation protocols for years.
I could not agree more. These two papers really represent a leap into the future for the field. But only time, or more importantly, new research will tell whether xenotransplants will be transplants for life, or they might serve as a better bridge to transplant in the future. So we stay in the era of organ compatibility or organ shortage. There is also a fascinating paper on ABO-incompatible transplantation. The authors used enzymatic conversion of a blood type A kidney to an O kidney during hypothermic machine perfusion. For listeners who may not be familiar with this technique, what exactly did the team accomplish and how could this reshape access to transplantation?
Yeah, certainly. So this is a very fascinating approach, I think. So a Chinese group used an enzyme, alpha-galactosidase derived from Bacteroides fragilis. And during hypothermic machine perfusion, they tried to convert a blood group A kidney into blood group 0. 3 hours after perfusion were sufficient to remove over 95% of the antigen A. And so the kidney was then transplanted in a brain-dead decent model without experiencing hyperacute rejection. That was one of the major challenges of these papers. So what's particularly compelling here is the single-cell sequencing data, which show the expression of accommodation-related genes, suggesting that the tissue was adapting, not only just surviving. This I think, is a very important step. If this translates to living donor setting, could fundamentally change how we approach AB0 barriers in organ allocation. And I think this is a very important milestone.
Yeah, I think so too. I mean, it's really amazing approach, and it might there might be no ABO barrier in the future, like using this approach. So one step forward. And now, I know this is close to your own research area. We move to allotransplantation. 2 papers in the New England Journal of Medicine made the editor's choice. One, demonstrating gene-edited immune-evasive human islets functioning without immunosuppression. And the other reporting the first in-human results of stem cell-derived fully differentiated islets. Antonio, these will probably be referred to as landmark papers later on. Could you walk us through these papers? Why these studies, why they matter so much?
Yeah, so this is my comfort zone, I have to say. So I'm very happy to discuss about it. But for allotransplantation, that represents an important step. Indeed, 2025 was, I have to say, very genuine landmark year for beta cell replacement. The first paper is from Uppsala team, and it demonstrated for the first time that the genome-editing human islet cells with HLA class 1 and 2 knockout and the CD47 overexpression is possible to achieve in order to evade the immune system surveillance. And these could survive. These islets actually could survive and function in human recipients without the use of the immunosuppression. And this is a very breakthrough and a proof of principle that in the field we achieved. 12 weeks of glucose responsive indeed, and insulin secretion and no rejection. That was the clinical endpoint achieved. This is a proof of concept, as I mentioned, that immune invasion through genome editing is clinically feasible. And the islet mass was intentionally insufficient to reverse diabetes, just to prove the effect of the genome editing strategy. And this was a safety and feasible study, but the principle is now established, and this can be applied even to the stem cell field too.
Okay. And the second paper, the Vertex trial?
The second paper reports the interim results of a phase 1/2 trial. This is a very challenging name, Xenotransplantation, stem cell-derived, fully differentiated islets in 14 type 1 diabetic patients worldwide. All 12 who received a full dose and completed 12 months of follow-up meet the primary endpoint, so the insulin independence, and 10 of 12 achieved insulin independence. So I have to correct myself. So only 10 to 12 achieved insulin independence. This is the first evidence that stem cell-derived tissue can function as an organ replacement therapy in humans. The limitation is that those patients are under immunosuppressive regimens and outcomes are comparable to the Edmonton protocol. But the supply problem is solved. You can generate a scalable source of stem cell. The next logical step is to combine these hypoimmune platforms that we have mentioned before with a limited cell sources to bypass, of course, the limitation in the beta cell replacement area.
That really sounds like the holy grail of tolerance. It would be amazing. And it feels like 2025 really made a difference. So 2 papers pointing at the same direction. So the same destination, but from different directions. And, well, the synergy could really be great.
Yeah, 2025 marked really a turning point for beta cell replacement with the first clinical demonstration that genome editing islets, as I mentioned, can evade immune rejection without immunosuppression in parallel. So in parallel, the Vertex trial showed that stem cell-derived cells can restore insulin independence, so effectively solving the supply chain problem. From my perspective, the next challenge is translating this breakthrough insight in accessible therapy for everybody, which are limited times and building robust and scalable manufacturing process.
Thank you. So, of course, all the other papers in this editor's picks are equally exciting and have major impacts in their fields. But discussing everything here in detail would probably last for hours. And I really, for our listeners, go to the Transplant International homepage and check out our selection. It's also the references to all the original papers. Another important field, machine perfusion, also is featured in our February issue. So, the machine perfusion is also featured in the top 12, but also transplant oncology, infectious diseases, transplant immunology, some new band classification updates. But maybe, Antonio, could you tell our listeners a bit more about how were these papers selected for the editorial?
Yeah, absolutely, Nina. So this was really a team effort. Indeed, we work closely with our deputy editors, fellows, and associate editor, gathering their input and perspective to identify the paper that truly moved the field forward and shape the narrative of 2025 regarding transplantation and all aspects, like, as you said, immunology, machine perfusion, and we shaped this fantastic report.
Thank you, Antonio, and also thank you to all the editors and the whole editorial team of Transplant International. So to all our listeners, you should really check it out. And now it's the time, as I said before, to also highlight our amazing February cover. And then it will bridge to the second topic we're discussing today, machine learning, big data, AI. So Transplant International has just published a narrative review on machine learning applications, specifically in lung transplantation. How does it connect to what we have already been discussing today?
I mean, this is very directly connected. Several of the advances we have just described, from machine perfusion, optimization, PGD prediction, and CLA-day phenotyping, are areas where machine learning is now being very actively applied. So this review from the authors gives a clinician a conceptual foundation for understanding the methods that machine learning is applied, and a clear-eyed assessment of where they have delivered results and where significant limitations remain in this field.
So it really hit the clinics. And for listeners who are new to this area or not so firm, so how would you frame the machine learning landscape as it is today?
I have to say that also for myself it's a little bit complex, but I will try to summarize in a better way. So the most fundamental distinction is between supervised and unsupervised machine learning systems. So supervised learning uses labeled data, patient with no outcomes, to train an artificial model intelligence to predict those outcomes in new cases. Unsupervised learning finds structure in a doubt without predefinitive labels, useful for identifying novel patient subgroups or molecular phenotype. Within supervised learning, the methods range from decision tree, intuitive and readable, through random forest and gradient boosting, which are more powerful but less transparent to deep learning, which can capture very complex patterns but still require large data set, and is essential a black box without additional explainability tools. So it's very complex.
Yeah, it's really very complex, and I'm still struggling to really get the point of it, or like a way of understanding the outcomes and how to use it. Where has machine learning shown the most convincing results? I mean, here for this paper and lung transplantation specifically?
Yeah, so the outcome prediction is where the most consistent evidence lie, actually, in this setting, particularly for survival and primary graft dysfunction. So random forests have repeatedly outperformed traditional Cox regression, detecting nonlinear relationship and interaction that linear models miss. So a particularly useful development has been SHT, shaped additive explanation, which quantifies each variable's contribution to model's prediction. In one study, this SHAP, I invited the reader to go in the paper and see, identify FEV1 and hepatitis B surface antibodies status as a top predictor of long-term survival, association that would likely have been missed conventionally in the regular analysis.
The primary graft dysfunction prediction seems very promising and an actively pursued approach, as it remains one of the most challenging early complications, also affecting long-term survival. It is indeed really important. And what are machine learning models capturing that our traditional approaches missed or could not detect until now?
It's one of the most productive areas, in my opinion. Indeed, Fessler and colleagues developed an innovative approach for incorporating data progressively across 12 perioperative stages. So the model's risk estimation is updated as more clinical information becomes available. And the Michaels et al. Group built a model using only pre-transplant variable released as an open access web calculator. And CI et al. Tested 9 algorithms in 802 patients with random forest performing best. What is encouraging in this overview is that these studies are covering on overlapping predictor sets, which adds on biological credibility.
Encouraging indeed. And what about organ allocation? Are we getting closer to seeing machine learning-driven allocation tools used in clinical practice?
Machine learning offers something that current lung allocation scores cannot. So the ability to model the donor-recipient pair as unit. Zafar and colleague developed and modeled LAPT, so the Lung Transplantation Advanced Predictor Tools, this is the acronym, based on over 50,000 cases, predicting 1, 5, and 10 years survival for specific donor-recipient combination, available as a web tool. So InsightX, that is the tool, integrates ex vivo lung perfusion dataset to assess lung sustainability and quantitatively. But I want to be balanced. So Brambat et al. And Dalton et al. Both found that machine learning did not meaningfully outperform standards model for post-transplant survival prediction. So we are not yet at the point in which machine learning demonstrability improves allocation decision in real-world practice.
Thank you. So I think my opinion on this is that, all this is depending on how well we train those machines, how well we report the data. So it's like garbage in, garbage out. So we really needed a better system that would then also be good for all the studies. So everything is streamlined, like you could compare between hospitals and all those scientific stuff.
Yeah.
What about imaging and omics? Also they seem like natural domains for machine learning because it's huge data sets.
Yeah. So both illustrate what machine learning can do that human analysis cannot. So in imaging, quantitative CT with support vector machine has achieved over 85% accuracy in predicting bronchiolitis, for example, syndrome using just 3 parameters. The Caliper platform can phenotype CLAD and predict graft failure reproducibility without expiratory CT. So in omics, on the other side, Halloran and colleagues have shown that molecular profiling of the biopsies classified as rejection more coherently than histological alone, and then molecular T-cell mediated rejection scores predict future graft loss. There are also early work on volatile organic compounds profiling from BLA fluid as a non-invasive diagnostic approach. The consistent limitation is small data set, often fewer than 100 patients, and almost no external validation. And I think this is the only limitation. So when you have a very small group of data, absolutely, this is a bottleneck, I think.
Okay, yeah, that sounds logical. And one of the cases where size is really important. But there are other barriers for clinical routine adoption, right?
Yeah. So they are slightly consistent across the literature. So data set size is a primary constraint. Most studies are based on a single center cohort of 2,000 or 600 patients. That is quite insufficient for deep learning in particular. So cross-center inconsistency in protocol and definition limit the possible generalizability of the data. So reporting quality is the real problem. Many papers provide very limited information about model architecture, and hyperparameter choice, making replication essentially impossible. The authors call explicitly for adoption of MI claim and TIPOID AI reporting frameworks, and interpretability in terms of that a model that a clinical cannot interrogate will not be adopted in the clinical routine, so regardless of its discriminative performance.
I see. So the reporting, working together in this field is, though we have the machines that help us, is really the thing that's most important.
Yeah.
And finally, when looking ahead, what should the field prioritize going forward, in your opinion?
Yeah, so take all this information together. I think that multi-center collaboration above all, so having larger, more diverse prospectively collected data set with, most importantly, harmonized definition is the key. So true external validation should become the minimum standard. On the methodological side, there are underused approaches like stacking ensembles, generative adversarial networks for data argumentation, and transfer learning for data scarce setting. But the overarching message is the right one. So machine learning should complement clinical decision making, not replacing at all, and the clinical judgment. Its success will depend on the true collaboration between clinicians, data scientists, ethics, and also regulators in order to have a good interpretation of the data.
Thank you for making this point. So I really have to stress it should complement clinical decision making, not replace it.
Yeah.
And now, stepping back across everything we already discussed today, what picture emerges? So can you give our listeners a take-home message today?
Yeah, so I think that they tell a coherent story. So the top 12 papers show us that fundamental biological barriers of transplantation, like organ viability, immune rejection, compatibility, are being dismantled simultaneously through genome editing, stem cell, and machine perfusion. On the other side, machine learning system review show us that the methodological tools to navigate and optimize this complexity are maturing, and even if they are not yet fully ready for routine clinical deployment, still they are improving really in a very strong mood. So we are in a genuine transformative period, and the advances are interconnected from each other. So the combination of gene editing, for example, stem cell-derived islets that evade immune system, machine learning optimization, donor-recipient matching with machine perfusion organ are where the next generation of breakthrough will come from, for sure.
Thank you, Antonio. It was a pleasure. This has been a rich and really wide-ranging conversation today. Thank you so much for sharing your insights.
Thank you, Nina. Both papers are open access in Transplant International, I want to remember, and I encourage all the listeners to read them directly and take also the direct overview about these two papers.
So this was it for today. Thanks to all of you for joining us for this episode of Transplant International Podcast, where science, innovation, and community meet. And please don't forget to subscribe. We'll be back soon with more conversations from across the field of transplantation, and of course with our next regular episode for the March issue. Thank you and bye-bye.